Hello everyone, I'm Ms. Singyao. I hope everyone is doing good at home. Today we will be taking class on biology on the topic reproduction in living organism. In our everyday life, we have seen a countless number of plants and animals species, all of which have a limited lifespan and they are bound to die. Yet, all these species, they are existed for several thousands of years. And how is this possible? That's because the living organisms, they reproduce. That is, the living organisms give rise to young ones, and this in turn, they grow and reproduce again their own kind of the species. So this ensures the survival of the species for generation after generation. So let's see the definitions of the reproductions I have given here. It is a biological process which is an organism give rise to young ones, that means offsprings of the same species. So this is one of the most important characteristic features of the living organisms, and it is the only way by which the living organisms multiplies and give rise to new organisms. Let's see the purpose of the reproduction. The main purpose of the reproduction is continuity of the species, population organization, variations, and life. All the living organisms, they have a definite lifespan, and all are not more immortal. So some of the organisms, they have a limited lifespan of just a few days, few hours, and some are several thousand years. Let's take the example of a mayfly. Mayfly live for only one day, and human being 100 years, cat 25 years, dog 25 years, and people tree 200 to 300 years, and then red tree 2000 to 4000 years. So in order to continue this, reproduction is the only way to maintain this continuity of the species. Living reproduction maintains all the population organization of the living organisms, like the young ones, aged ones, and the other ones. In order to maintain this, reproduction in living organism is the only means. During the reproductions, the genes of the parents are kept mixed with one another. That means transmission of the genetic materials are evolved in the reproductions. So this arises the variation in the offsprings. So in order to maintain these variations and help in the evolution of the species, this reproduction is very important. So that is the purpose of the reproductions, and all the living organisms won't be survived if there is no reproductions. Now in our living organisms, we mainly see the two types of reproductions here, asexual reproduction and sexual reproductions. So in the coming lectures, we will discuss step by step, and first we'll go to the asexual reproduction. So let's see, what is asexual reproduction here? When we talk about asexual reproductions, we mean the production of the offsprings from the single parent, that means uniparental reproduction, uniparental, only single parent. And here, it is only from the single parent, that's why they don't need to form the zygotes and they don't need the formation of gametes, only one parent. So, all the offsprings which are produced from these methods are all identical. Not only identical, but they are exact copies from the parents. That's why all the individuals of these asexual reproductions are termed as a cloned. Why cloned? Because morphologically and genetically they are exactly with the parents. That's why we also call as a cloned. And in this asexual reproductions, only the mitotic cell division is taking place. So the name we call it as a somatic reproduction. Some of the few characteristic features of the asexual reproductions are given here. They are single parents, and gamete formation is not required, and there is no fertilizations, only mitotic cell divisions, and daughter organism produced are genetically identical with the parents, and here the multiplication will goes rapidly. So these are the main characteristic features present in the required in the asexual reproductions. This asexual reproduction usually occurs in the unicellular organisms like the monerans, protists, and plants, and in certain animals. So some of the common methods of the asexual reproductions are given here. Number 1 is fission, number 2 budding, number 3 fragmentation, number 4 sporulations, number 5 is vegetations. We shall go one by one in details. First, we will see the fission reproductions. So this fission here, the parent body is divided into 2 parts, and these individuals are exactly identical to the parents. So 2 types are here in the fission, that is binary fission and multiple fission. So first, let's see what is this binary fission. So binary, from the word binary itself, we mean 2. So the parent bodies will divide it into 2 equal halves, and each of these 2 halves will form into another 2 independent organisms when they get mature. So based on the divisions of the plate, these binary divisions, they are again classified into 3 types: irregular or simple fission, longitudinal binary fission, and transverse binary fission. So here, and these are the diagrams for each of the fissions. So binary, in the irregular simple fissions, the division can be taken place in all the sites. That means this is the parent. Example, this takes place in the species amoeba. Okay, so amoeba here. Division of the parent body can be through any plate. So this is the body of the amoeba parent body. So this division can be done through this way, through this way, through this way, through this way. So any plate, the division can be done. And first they will divide the nucleus, and after that they will divide the cytoplasm, cytokinesis, and then they reproduce into 2 daughter cells. That is in the amoeba. So in amoeba, simple fissions takes place. In the longitudinal binary fissions, we will see here in the euglena. In the euglena, the parent bodies are directly divided into longitudinal axes. Here, longitudinal axis, they will divide first the nucleus, and after that they will divide the cytoplasm and give rise to these 2 daughter cells. In the transfer again, binary fissions, this one you will find in the Paramecium species. So here, they divide the species in a transverse way. Here like this. So first they will divide the nucleus which are present in the parent body into 2, and then they give rise to 2 daughter cells in a transverse axis. So these are the types of divisions in the.
Yes. So.
He flips away. Yeah. Okay, next. Now we'll go to the multiple fission. So from the word itself, multiple, it means more than 2. So in the multiple fissions, the parent divides into many daughter organisms. So here the nucleus of the parents divides repeatedly, and they form a number of daughter nuclei. And these are found in the Plasmodium species, and in the amoeba it is only under the unfavorable conditions. So here, this is the parent body of the Plasmodium. They divide the nucleus and they give rise, repeatedly divided the nucleus, and they give rise to many daughter nuclei. Next is budding. So budding is also a type of asexual reproduction. Here in this, a small bud, or we call it as a small outgrowth from the parent body, develops, and this bud, when the cats mature, they detach from the parent body and they develop into new individual organism that we call it as a budding. These budding are mainly found in the yeast and hydra. So this is the parent body and small bud is developed in the parent body. Later on this bud will detach from the parent body and they develop into a independent new daughter individuals. Same thing in the hydra also. This is the parent body, the small outgrowth of the bud. Here they grow in size, the cats mature, and when the cats mature, they detach it and they develop into new daughter cells. Next is fragmentation. Fragmentation is also a type of asexual reproduction. So here, fragmentations, mainly the parent body are breaks into many fragments, and each of these fragments develop into a new daughter organisms. So this you will find in the spirogyra. Next is the sporulation. So sporulation is also a type of asexual reproduction, and these are found mainly in the plants like bacteria, fungi, and algae. And the formation of the new species are mainly from the spores. So spores are nothing but they are the minute single cell reproductive units, and after maturation of these spores, they grow in size and they detach from the parent bodies when unfavorable conditions comes. So these are found, they are locked inside the sporangium pores. These pores, minute pores, they lock inside the sporangiums, and when the favorable time comes, they dehisce and they detach from the parents. So this type of the reproductions you will find in the penicillium and the mushroom. Next is the vegetative propagations. So vegetative here, this is the formation of new plants from the vegetative units. So when we talk about vegetative units, it is the parts of the buds, rhizomes, and tubers. These vegetative parts, we call it as a vegetative propagules also. So mainly in two ways, it propagated in two ways, natural and artificial propagation. So in the natural here, these plants multiply naturally, and the vegetative units such as the buds, tubers, rhizomes, they will detach from the parent body, that means from the mother, and develops into new independent plants under suitable environmental conditions. These are mainly done by the following methods here. Written down here. Number 1 is root, number 2 is underground stem, 3 creeping stems, number 4 is leaf, number 5 is pupils. So roots, both the taproots and adventitious roots are used for the propagation of the plants. And then underground stems, all types of the underground stems are here. Tubers, example potato, bulbs, garlic and onion, etc. Corms, colocasia, crocus, rhizomes, banana, ginger, turmeric, suckers, mint, chrysanthemum, etc. So this can be propagated through this, and potato is only produced by seeds. Clipping stems, these are the stems, but they are weak stems. So the stems usually they are slender and they are horizontally branched, and at interval of the time they develop the rhizomes and they propagated in that way. So especially centella, strawberry, water hyacinth, oxalis, xanadont are all propagated by this method. And the leaves, many of the leaves have these adventitious buds, and they can be cut and help in the vegetative propagations. Begonia, bryophyllum, and pupils, pineapple, etc. So all these are the types of the vegetative propagations which can be done to reproduce the new species. Number 2, that is artificial or horticultural methods. So these are the man-made technique propagations which is mainly used for propagating the desired varieties according to the human requirement. So nowadays, many of the horticulturalists, they propagate the ornamental plants in this method. So various methods are used. Number 1 is cutting, number 2 is layering, number 3 is grafting, number 4 is micro propagations. So here are cuttings first. Cuttings are cut pieces of the stems, roots and leaves, and later on these are planted into the nurseries. So for this purpose, we use the root promoting chemicals such as IPA and naphthalene is used. So the best season for the cutting propagation is early spring or during the rainy season. So in the root cuttings, this snake plant, Bryophyllum, Begonia, all these are used by root cuttings. So here the cut roots of 20 to 30 centimeter length are dipped in the hormones and later on they planted into the nursery. So in that way, this snake plant, Bryophyllum, Begonia can be propagated. Stems, same thing here also. 20 to 30 centimeter long of the pieces of 1 year old are cut it out and they put into the chemical hormones. That means IPA, maybe IPA or it may be a naphthalene acetic acid, and it can be planted into the soil. So China rose, very commonly we use China rose, bougainvillea, sugarcane, coffee, tea, citrus trees, all these are used by these stem cuttings. In the leaf cuttings also same thing. Leaf, they cut into various pieces in transverse way, and then before planting into the soil, we take the cut pieces of the leaves and dip into the root promoting chemicals and later on place in the vertical positions and propagate in that way. Next is the layering. Layering is also an artificial method of propagations. Here mainly the root cuttings are involved. That means the plants used for propagations, especially it should be a diploid or gymnosperm plants which have a cambium. So here adventitious roots are induced to develop on the soft part of the plants while they are still attached to the plant. Okay, so two methods: simple layering, that means small layering, and air layering can use here. So simple layering here, layering here. The middle portion of the part are removed. The middle portion, the bark of the middle trees. Here we will remove the bark or we defoliated the cover, and then we add the hormones here for inducing the roots. Okay, and later on, after 2-3 months, okay, after 2-3 months, all the adventitious buds will start developing here. Then we can cut it off here and separate it from the parents. Then directly we can plant this into the soil. And in the air layering also same thing. We covered with a plastic, we call it as a grafting clay, mixed with the cow dung, and then root hormones are all filled with this and covered with the plastic. And after that, the root will start develop in these portions. So when they develop these roots, we can cut it below the plantage here and separate it out from the parents. That's how we do in the layering. So next is the grafting. Grafting is also a vegetative propagation method. Here, two separate plants are joined together and they form only one. That means it is a special technique that vegetatively join the two plants into ones. So here, dicots and gymnosperms which have a cambium ring are done by this grafting. Types of grafting are here: dung grafting, crown grafting, wedge grafting, and approach grafting. Next is the micro propagation here. Micro propagation. This is also called as a tissue culture. Why? Because in these propagations, mainly the culturing of the cells, tissue organs in the free environment germ cells. So the plants which are produced from this method are old virus-free plants, and they are the clone of the plant from which the plant was obtained. So growth regulators such as the auxins, cytokinins, gibberellins, and then other hormones are supplemented in the medium. So these are the various methods of the asexual reproduction in the living organisms. We'll see the now advantages here. Some of the advantages of the vegetative propagations you will find here. In the seedless plants, propagation can be done, or in the seedless plants. Only the method of propagation is through this vegetative, okay? And then sugarcane, bananas, seedless grapes, seedless oranges, all these are done by vegetative propagation. And number 2 is plant can be retained and multiplied indefinitely without any change or variations, and good quality of the fruits and flowers can be produced in these vegetative propagations. They show a genetic uniformity because there is no mixing of the genes, and rapid multiplications will be there. And here, disease-free plants can be produced in a short period of time. Survival of the species also 100% compared to the seeds, and all the genetically modified plants can be produced by using these tissue cultures. Some of the disadvantages are there, and here number 1 is the vegetative propagules, that means the stems, the rhizomes, the bulbs can be easily decayed and can be prone to viral bacterial and fungal disease. So that is the one advantage. And number 2 here, in the vegetative propagation, since there is no mixing of the gametes between the parents, no variations are exhibit here. That's why the plants will show degenerations and they are less adaptable to the changed environment. And these propagules did not disperse by nature, therefore they will cause the overcrowding. So these are some disadvantages of vegetative propagation. Now here, as a whole of asexual reproductions, disadvantage and advantage. Number 1 is advantages. In asexual, since the reproduction is only from the single parent, they don't require the mate. Okay, so they don't require the reproductive organs for male and female. Uniparental reproduction, so no mate is required. That is the advantage number 1. And number 2, it is a very quick mode of reproductions. Within a short period of time, a large number of the organisms can be produced. Number 3, single parent may produce a large number of the offsprings. And number 3, the offsprings and the young ones or the individual ones which are developed from these asexuals are all genetically similar to their parents, and this is the advantage here. Since there is no mixing of the genetic materials, they will not have the variation among the species. Number 2 is due to rapid multiplications, overcrowding of the species can occur. And number 3, organisms will have a low adaptability to the changed environment. So these are the various disadvantages and advantages of the sexual reproductions. Okay, thank you.